Okay, I am Dr. Greg Nacarelli, and today I'll be talking about methylation, artificial intelligence, and metabolomics. What is methylation? So this is the process of adding a methyl group, that being a carbon and three hydrogens, from a methyl donor to a methyl acceptor. Examples of methyl acceptors include DNA, neurotransmitters, proteins, waste substances, homocysteines, vitamins such as methylated folic acid, which is probably the most infamous one, and histamine. It plays a crucial role in many different bodily processes, blood vessel health, fertility, embryonic development, cognition, cellular and liver detox, creatine and choline production, just to name a few of the items. Overall, methylation it's very complex, it's tightly regulated, especially DNA methylation, which is being shown on the right here. So methyl groups will be attached to our DNA to affect the expression of them. Usually methylation of DNA means to silence a gene, but not always. When we refer to methylation pathways, we're really referring to one carbon metabolism, one carbon being that methyl group, which contains one carbon. This breaks down into three different main areas. Well, it could break down into several different areas, but the main areas I'll cover today is the methionine cycle. So this includes homocysteine being converted into methionine, which then gets converted into S-adenosyl methionine, which then gets converted into S-adenosyl homocysteine. And then there's the other part of the cycle, which is the folate cycle. This is the part of the cycle that actually generates methylated folate. So with this, we have folate being converted into tetrahydrofolate. Tetrahydrofolate is converted into 5,10-methyl tetrahydrofolate. And then with the help of the MTHFR enzyme, is converted into 5-methyl tetrahydrofolate. And so we have production of basically this methylated folate. We also have homocysteine being converted into methionine. Homocysteine is the one that's most notoriously known for if this is elevated, there are negative consequences to that. It's most directly associated with aberrant methylation, and it's associated with atherosclerosis and cardiovascular disease and other health conditions I'll mention later. The idea is that if homocysteine is elevated, then there's something going wrong with this or maybe even this. And so how do you figure that out? How do you tease that out? So the genes associated with methylation, there are many. A lot of people just focus on this one. Some people will just look at this one and help to figure out if methylation is aberrant. But really, if you're trying to be comprehensive and look at the entire gene pathways that can affect methylation, you want to look at all the different genes. And so some notable ones, there's methyl tetrahydrofolate reductase, which is methylene, and that is this one. So that converts basically 5,10 tetrahydrofolate into 5-methyl tetrahydrofolate. There's MTR and MTRR, which are essentially the gatekeepers of converting homocysteine to methionine. They require B12. There's variants in those genes where they could not function well. Also, if there's a high degree of oxidative stress or inflammation, it could also impair these genes, and they require B12. So there's lots of different factors that, aside from MTHFR, could block that conversion pathway from homocysteine to methionine. SHMT, serine hydroxy methyltransferase, which is somewhere over here. So it's basically functioning to alternate between serine and glycine, and then functions in the tetrahydrofolate metabolism. TYMS, thymidylate synthase, it's down here. So this is getting into nucleotide synthesis. So that's another aspect of methylation, is that it will be helping to create nucleotides, that being the backbone of our genes. DHFR, dihydrofolate reductase, should be, okay, here it is. So yeah, folic acid from our diet or from synthetic folic acid will have to go through this enzyme to make dihydrofolate, which then will further go through the rest of this cycle. So that's a big one. Methylene tetrahydrofolate methylene tetrahydrofolate dehydrogenase, MTHFD1, which is here, again, part of this folate production cycle. Also, betaine homocysteine methyltransferase, BHMT, another important one, will serve to do this alternate pathway. So some people will just attribute homocysteine only gets converted to methionine via this part. Actually, no, it could go through this cycle, which is where we need adequate choline, which will get converted into betaine, which will get converted into dimethylglycine, and that conversion process facilitates the conversion of homocysteine to methionine. So as I'll go through later, having adequate methylation isn't just methylfolate. There's many other factors that are attributed to this, not just genes, but also dietary products that are affecting methylation as well. These are just the genes. I'm not covering the risk variants in the genes. There are many. That will be a future topic that I could potentially cover. But for now, I'm just looking at the genes, or specifically the gene products, which are these. So every one of these is basically a gene coded for a protein So MTHFR is a protein that was made from a gene, and subsequently the same thing with all these. But I'll cover the genetic variants in these future lectures. What are the metabolites associated with methylation? And so with the metabolome, and if anyone's familiar with Theriome, we don't just look at one metabolite and attribute all health associations with one metabolite. You have to look at the metabolome, which means you have to look at different areas of metabolomics and how they're associated with each other. You could kind of break it down into several main different categories here. And so within this methylation cycle, methionine, homocysteine, S-adenosylmethionine, S-adenosylhomocysteine, there's this offshoot of the methyl cycle called transsulfuration. So homocysteine can get converted into cystathionine, cysteine, glutathiones. These are other metabolites that can be measured. There's within the folate metabolism, obviously methylated folate, but also things like glycine, serine, sarcosine, choline, betaine, other metabolites from the methyl cycle going into nucleotide metabolism. You can look at something like adenosine. So, and purines are a type of nucleotide. And then also polyamine production, so putrescine. This is associated with gut function as well, so you could measure this as well. So there's a lot of different metabolites that you could look at to try to get an association with how's a methylation cycle working. Just looking at homocysteine, there's just so many other metabolites that could be implicated. So, you want to look at the whole picture. What are the health implications of methylation imbalance? So atherosclerotic cardiovascular disease, cancer, dementia, gout, anxiety, depression, miscarriage, oral cleft malformation. Sometimes there's inborn errors or severe methylation defects where you'll see these kind of malformations that are born congenitally. Autism, adverse drug and environmental reactions, and this is just to name a few. There's quite a bit of health conditions, not solely caused by methylation defects or imbalances, but rather they contribute to these health conditions. So when we look at the diet, how do we support a diet, or how do we support methylation with a healthy diet? And so general dietary supporting methylation nutrients include dark leafy greens, beets, eggs, oily fish such as salmon, legumes, liver, cruciferous vegetables, okra, mushrooms. Some nutrients that support methylation include, protein. That will include arginine, cysteine, methionine, tryptophan, tyrosine, biopterin, creatine. Fats, DHA, phosphatidylserine, choline. Minerals, magnesium, zinc, phosphorus. Vitamins, so these activated forms of these B vitamins, including active or methylated folate, methylated B12, niacin, riboflavin, active B6, P5P. Essentially, every B vitamin, for the most part, has some type of entrance into the methylation cycle. So just overall B vitamin sufficiency in general. Some dietary considerations. I thought that was really interesting, but they've looked at just methylation, methyl donors, methylation metabolites, comparing seasonally. When you look at the rainy season from July to September, plants just make more nutrients. They have higher chlorophyll. And so they're higher in riboflavin, folate, B6, and methionine, these crucial nutrients for methylation. So in rainy seasons, you see better methylation or methylation sufficiency versus dry season, that being from February to April. Assuming you're eating seasonally or locally, and you're in the part of the hemisphere where that is colder and drier, you'll see lower folate, riboflavin, B6, and methionine. So there is this kind of seasonality between methylation sufficiency versus insufficiency. There are many different types of folate, so just to cover this briefly, there's folate, which is B9. This is required for numerous processes in the body. It's an essential vitamin. Then there's folic acid. This is the synthetically created folic acid. This is in most, like either prenatal supplements or vitamins, at least ones that don't attempt to include the activated or methylated folate. The issue with this is it can block the dihydrofolate reductase receptor or enzyme, and that can obviously lead to more consequences in our body's ability to make this methylated folate. So generally, we recommend for people to avoid this synthetic folic acid. There's then folinic acid. This was originally created as a drug, leucovorin, to help combat the side effects of methotrexate, which is a cancer drug that in itself is a dihydrofolate reductase inhibitor. And so this is one supplement some people will take. It's a bit upstream from the next one, which is L-methylfolate, which is down here. There's two main versions of this. There's the calcium L5 methyl tetrahydrofolate, known as luvomifolaate calcium, which is a direct bioavailable form of folic acid. And then there's this one where I feel like most supplements are using this form nowadays. It's called quattrifolic. So it's 5-methyl tetrahydrofolate bound to a glucosamine salt. And by doing this, they've made it highly bioavailable. So these are the main ones that are currently on the market with large fluctuations in dosing, and that's a whole other conversation. So what can deplete us of methylation or methyl donors? So if you are deficient in any of the items on the previous slide, so a methyl donor deficient diet, that will obviously deplete you. If you have genetic predispositions towards utilizing or making methyl donors, that will also deplete you. That goes back to the genes on the other slide. If you have excess niacin or excess nicotinamide, this will decrease betaine, which through that other pathway I showed will ultimately increase homocysteine, so that's not good. If you have low folate specifically during pregnancy, where you have increased folate needs, that's also going to deplete you of methyl donors. If you have a high-fat diet with low B12, this is going to decrease the enzymes in the methyl donor pathway, so that's not good. Nitrous oxide gas, think dentists, this will impair B12 metabolism. And so there's actually a good amount of people where when I think somebody's gone to the dentist and they've had nitrous, the first thing I think of is methylation dysfunction because they're going to get that B12 impairment that's going to block those MTR, MTRR enzymes. Chronic stress. So within methylation, we could talk about what it's doing to different individual enzymes and breaking down neurotransmitters and histamine, things of that nature. But it's also needed for making marks on our DNA. When you look at these like methylation aging tests, and they're looking at methylation marks on DNA, one of the, at least like 30% of all the genes that they're looking at for these methylation changes are in genes associated with stress. And so you're going to change the whole methylation landscape of your DNA by being chronically stressed. And so, and not only that, it's going to deplete you of methyl donors in the process of being stressed. So stress is probably one of the biggest ones that one needs to target in terms of just supporting methylation. Toxins and toxicant exposures, toxins being naturally derived toxins in our environment like venom, and then toxicants being man-made pollutants or chemicals. So we have mycotoxins being toxins, and then organic pollutants such as BPA. We also have heavy metals. So any of these things can put a stressor on our methylation system because then we require more methyl donors to get rid of them. One thing that is interesting is that going back to, actually I'm going to bump up a couple slides here. You see this thing DNMT. So this enzyme will basically add methyl groups to our DNA. So that's something to also consider when we're talking about, okay, what is our body using methyl groups for? This enzyme is inhibited by EGCG, one of the active compounds from green tea, resveratrol, genistein, which is from soy, and then alpha-ketoglutarate, which has had a lot of notoriety in recent literature for being an anti-aging molecule. And so typically people associate more methylation marks on the DNA with aging. Now, again, it's not a hard or fast rule. So the question then becomes, okay, if we're inhibiting these DNMT enzymes, are we actually decreasing aging? It's because typically we think of these things as anti-aging molecules. So just another wrinkle for this methylation conversation. Okay, so what does it take home for methylation? So this is a very tightly regulated process. There's lots of players going on. It plays a role in numerous areas of the body and health. When, as a practitioner, people are coming to you, usually it's not that they have a methylation problem. There's usually some health condition that they have of which methylation is a contributing factor, not the sole cause of their health issues. So it's just one aspect to address. Methylation can be optimized through sufficient dietary intake of methyl donors, stress minimization, and environmental pollutant avoidance. You can assess your genetic predispositions for methylation status with a genetic test. And then you could look at metabolites associated with methylation to see, okay, what is the output of my methylation pathway? One thing to know is that you can have methylation dysfunction without genetic risks. And so the only way you would really know that is if you were looking at the metabolites, because like I said, you could not have risk variants in those genes, but you've been exposed to nitrous or you've been exposed to toxicants or you're vitamin deficient in general. So it is very important to look at the methylome or methylation metabolites. And we are proud to announce that Theriome is releasing a methylation index. And so from metabolites associated with methylation, we will create a methylation efficiency index score. And so we'll let you know if your body's ability to methylate overall is either poor or normal or optimal. And we will have recommendations to help support that littered through the report in the various different health domains. So this is something that we will be releasing shortly. So this is something to explore if you want to figure out your methylation. Okay, let's talk about artificial intelligence. Theriome utilizes artificial intelligence in its report creation. So what is this? So let's just start bare bones. Artificial intelligence is essentially, it means to incorporate human behavior and intelligence to machines or systems. In a way, we've kind of always been doing this, like when we have just individual markers and we do a calculation to micrograms per liter or something, right? This we're using a computer to calculate it. When we're branching out and we're finding extrapolations between metabolites and then between health states, it's where it gets very large, and we're using artificial intelligence to find those associations. Beyond that, there's machine learning. So this involves using methods to learn from data or past experiences, which will then automate the analytical model building. And so it's kind of learning from data sets. And then beyond that is something called deep learning, where we're getting more complex, where we have multiple layers of this, neural networks and processing. So this just kind of gets deeper and deeper in terms of the artificial intelligence to be able to learn from data. What does Theriome use in terms of artificial intelligence for metabolomic analysis? So machine learning. Again, this is where we allow the AI to learn and improve from data without being explicitly programmed. We then do network analysis. This is where we examine and elucidate the relationships between individual and groupings of data points. This is what's shown on the right here. And so these are metabolites that are associated with cardiovascular disease and atherosclerosis, which is kind of in the middle here. All these lines here are showing the associations between the metabolites and atherosclerosis. The solid line is a direct relationship. The dotted line is an indirect relationship. And then we could see whether it's a direct association or an inverse association. And so this kind of gets all over the place, right? This is where we need artificial intelligence to do these calculations and extrapolations. Okay, this is all well and good, but how does this translate to a patient visit? How do I know from which ones are high or low that I need to focus on? And this is just one health condition. No one just has one health condition. People have a lot of different things going on. So how is their atherosclerosis risk associated with their inflammation risk or their mitochondrial function, right? So when it gets to these layers of complexity, this is where the artificial intelligence can help us to figure out the next best steps forward. Which then brings me to digital twinning. So this is where we use artificial intelligence to create a virtual replica of a physical entity, and then we use data points to connect the two. And this is another aspect of what Theriome uses for its recommendations. And I'll be talking about that shortly. So how does Theriome use AI? So one, we have the data. We're using molecular biology techniques to get the data. We're using artificial intelligence and machine learning to kind of create the data. We have a bioinformatics pipeline where we're pushing the data through. And then we use AI models to or we train them to identify patterns across our 126 profiled metabolites. We have 12 health domains where we're looking at to create a personalized report. And this is essentially the backbone of interpreting complex biological information and converting it into actionable health metrics and recommendations. So first thing, we use artificial intelligence algorithms to identify key metabolites that are most relevant to health domains. We then use pattern recognition, and we do this by using machine learning models to train and recognize the metabolomic data that correlate with known health states. We then aggregate the key metabolites into a health domain. And then we use artificial intelligence to assign scores to a health module, which can indicate whether there's poor functioning, normal functioning, or optimal functioning of a health domain. These models are then tested via simulations to ensure accuracy. And then based on that performance of the models, we fine-tune them and retrain them to improve predictions. Overall, this leads to a continuous learning where the models undergo further training and enhancing their precision and adaptability with their predictions. So how is this incorporated into our reports? And so we need to interpret the model. So we have predictors in the report. How do we know which ones are most important? What are the relationships among the predictors? And how does a predictor affect the predictions? And in terms of interpreting our predictions, what are the most influencing predictors for a given prediction? And then what are the most actionable? And so this is where the digital twinning can come into play in order to discover this. So for digital twinning, this allows practitioners and researchers to visualize complex physiological processes. It offers a platform for simulation and testing of various health interventions, and then it serves as a predictive tool for health outcomes based on specific changes in lifestyle or treatment. And so what you're seeing on the right here is we have the individual and we have their digital twin, which is essentially a computer model. We have their health score on the right here, and as we kind of expose them to different interventions, you're seeing changes in the health score. And so a simplified version of this is, okay, you see one change, their health score improves, do another one, and then we've kind of hit this plateau where it's like, okay, what we're exposing the digital twin to is their health score is optimized, therefore these are the proper interventions that we should be recommending. What this actually looks like is this. So this is done 1,000 times. And so where the health score is kind of you're giving them interventions, it might increase it for a bit and then maybe decreases. But overall, we'll find the health interventions that are most fruitful for having the best health score compared to all these other digital twins. And so by doing this digital twinning, we're able to be more precise with our recommendations to shift the metabolic landscape that's ultimately going to create an improved health score. And I will be going over this a little bit more later on in this presentation. How does this benefit the practitioner? And so by doing this digital twinning, we have improved diagnosis and treatment. So we're able to understand health states more accurately. We're able to develop health protocols that are more precisely tailored to the client's unique biological profile. It improves efficiency, and so this reduces the time needed to analyze complex data, allowing practitioners to focus more on patient care. It allows for more proactive healthcare. So by identifying risks early, practitioners can implement preventative measures, potentially avoiding the progression of poor health states, and allows for enhanced patient engagement. So having this detailed yet comprehensive report, it fosters better communication between practitioners and clients, empowering the clients to participate actively in their health management. So it's by giving more quantitative data, we could kind of track progress, and especially doing this not just once, but over a period of time and seeing the health score change. It just provides a more real dynamic between the patient and their health versus kind of just a less quantitative or even qualitative approach to health management. So we talked about metabolomics, we talked about artificial intelligence and using that to look at metabolites or metabolome. Why should we look at it in general? By the time most people are ill, we've had shifts in our metabolome for quite some time. And shifts in metabolites, they're part of our body. Eventually, it's going to affect cells and tissues and then organs. If we look at the metabolome, we're looking way upstream before that happens. And so our ability to intervene before we see more end organ changes is just much sooner by looking at the metabolome. There's many different types of omes. There's the genome, which looks at our DNA, the transcriptome, which looks at our RNA, proteome, which looks at collections of proteins, the microbiome, which looks at our probiotics and bacteria, and then the metabolome, which looks at our metabolites that are part of metabolism. There has been a lot of research in metabolomics, also multiomics as well, especially since around 2016, research in metabolomics, or excuse me, multiomics has just skyrocketed. So it's an active area of research in terms of people looking at not just the individual omes, but how they're interacting with each other. And it's kind of the holy grail of precision medicine is just finding this data in order to overall improve health states. So why look at the metabolome? So if we're doing lifestyle changes, if we're doing dietary changes, if we're taking supplements, we're not going to see changes in the organs first, or even the tissues or the cells. The first thing we're going to see is shifts in the metabolites. And so the way we'll know how we're properly shifting the metabolites is really to measure them. So some people say, okay, is metabolomic genetic testing? No, it is not. They're distinct and different. Can I just do a genetic test and not the metabolomic test? The thing about genetic testing is while it can be predictive of health risks, it's not absolute. And so you could have a genetic risk for something, but not see the disease manifesting. Or you may not have a genetic risk for something, but you see it manifesting. And so how do you account for the discrepancy between the two? Genetics isn't operating in a vacuum. It's always affected by other things. It's affected by your diet. It's affected by your stress, your lifestyle, your environmental exposures. And so how does those affect the genetic expression? Well, the genetic expression ultimately would be reflected in the metabolome. So by measuring the metabolome, you're getting a more direct view of what's happening, for example, with methylation. If you just look at the genetic testing for methylation, you're not getting an accurate picture of what is your methylation output. So this is where looking at metabolites can be fruitful.